So, hi everyone, grateful to be here. I'm Bianca Bolocan, and this is a short history of CRISPR. I'm just going to pull up my little PowerPoint, and then we can get started. Yep, here it is. So, early genetic engineering before CRISPR was expensive and time-consuming. One such example would be zinc finger nucleases, or ZFNs. They are man-made combinations of zinc and protein that bind onto a specific DNA sequence and snip it in two places. They are extremely expensive and time-consuming, as I've said before, and another problem with them is that they were very likely to cause off-target edits. Another method includes transactivator effector nucleases, or talons, which are derived from certain plant bacteria. They did not really fix any of the issues with their predecessors. The first person to study CRISPR in depth was Francisco Mojica. He discovered a palindromic sequence separated by spacers within the bacterium Halopherax mediterraneae, and he copied those spacers, which seemed unfamiliar to him, into the BLAST gene database, eventually finding them to match with a phage infecting E. coli. Around the same time, French researcher Gilles Van Gogh and Russian researcher Alexander Bolotin also discovered the sequence. All three came to the same conclusion, which was that the sequence played a role in a bacterium's immune system, so to speak. Later on, the lab of Emmanuel Charpentier would perform a set of in vitro experiments to study how CRISPR works. And now would be a good time to explain that. So, a pre-CRRNA is copied from the CRISPR locus using only the spacers of it. This CRISPR locus being the part that Francisco Mojica discovered. Following that, a tracer RNA made around the same time proceeds to fold the pre-CRRNA into a CRRNA, which is then compared to the bacterium's genome by a compound called Cas9. If a match is found, the Cas9 cuts the DNA, rendering it harmless. Charpentier's lab found that the CRRNA and tracer RNA can be combined into a chimeric gRNA, which can then be used on non-bacterial creatures if a Cas9 compound is also introduced. A template piece can then be introduced alongside those two elements to allow any kind of edits or new genome to be introduced. Over time, CRISPR would go on to evolve in many different ways, designed earlier on mainly by the labs of Feng Zheng and Jennifer Duanda. Although nowadays research is conducted by various labs. Cas9 became more and more accurate. Multiplex genome editing has been invented, made specifically in Feng Zheng's lab, and different uses have been found for the technology in many fields. Here are a couple sources, if anybody wants to look at them, and a couple more. And finally, any questions?
So I have two questions from our judges. So what made you interested in investigating the history of CRISPR?
Oh, mainly I was just really interested in genetic engineering overall, and I kept really hearing CRISPR mainly as a buzzword, but I was really curious where it actually came from.
Got it. Yeah, I think in your presentation you broke it down in a way that was really easy, understandable. So what are the next steps for your research? Do you want to build upon this topic, or are you interested in pivoting to something else?
I was mostly interested now that I've really found out how it works to try and use it for myself and see a bit more of a hands-on approach to it if I can find one.
Very cool. Do you have any plans for those next steps?
Not yet, not really. I'll have to do more research.
Mhm. Was there anything unexpected or surprising that you discovered in your research?
Uh, sort of. I mean, I didn't really expect for a small rivalry to really form in the development of CRISPR.
Yeah, I'm sure that pushed the innovation. Um, okay, great. Thank you very much.
Thank you.